speaker is Regina Enninful. She is a PhD student at Kansas State University, and she will be talking about part of her project today.
Good afternoon. I would like to say thank you so much for the opportunity to come and share on my research work. And I'm also very happy to be in Florida because it's my very first time. So this afternoon, my work is on the characterization of parents of sorghum mapping populations exposed to water deficit stress during the vegetative stage. And for the most part of today in our symposium, I think the platform has already been laid that there is a climate change challenge. And most importantly, in relation to droughts, it's been noted that the global climate models has projected increased intensity and magnitude in water deficit conditions. And this is especially challenging for the arid and semi-arid regions of the world. Now, even if you come to the humid side or humid areas where there is higher agricultural productivity due to abundant rainfall, there can be periods of drought within the growing seasons, as well as unexpected years of severe drought. Therefore, drought or water deficit stress is a key abiotic stress that limits crop productivity, and as such, there is a need to find drought-tolerant plant traits. Now, talking about development of plant traits, there's been three main strategies that have been noted that plants use in order to cope with drought. The first is the avoidance strategy, where there is maintenance of high plant water status irrespective of the stress. And this the plant achieves by having a maximum uptake of water or minimizing water loss. The second is the escape strategy, where the plant induces and completes its life cycle before the onset of severe stress. And this that's by making proficient use of reserves. And then the third strategy is the tolerance strategy, where the plant is able to undergo its life processes using limited or under limited water availability. And this it does by making some osmotic adjustments and also scavenging for reactive oxygen species. Now, the approach to developing or improving drought tolerance involves basically one, discovering the existing drought tolerance mechanisms and traits in plants, identifying the genes involved and incorporating it into improved cultivars. These steps requires an understanding of the basis of drought tolerance in existing plant species. And I think Dr. Stephanie Green did a lot of explanation on this aspect of my introduction. So look at an example of a drought-tolerant crop, which we already know is sorghum. That is why my work focuses on that, because we want to know, it's already known to have drought tolerance ability. So then my work focuses on the three steps. The first aspect, where I'm trying to discover existing drought tolerance traits in the plants. And then my work basically tries to characterize sorghum, looking at the shoot, and also, very importantly, the root, because most research focuses more on the shoot than roots. That's on one editorial that I read. It's like comparatively more research is being done on the leaves, the flowers, the fruit, but then little is being done on the roots. And it's mostly, I don't know, because plants get themselves buried in the ground, and unfortunately, they get forgotten in research activities. But in my research, they are not forgotten. So I also look at the shoot and as well as the root. So on that basis, my research objective was to characterize parents of sorghum mapping populations for both the shoot and root-related physiological and agronomic parameters under water deficit stress. And my hypothesis was that water deficit stress during the vegetative stage will induce different drought tolerance traits, and the resultant changes in the physiological and agronomic traits among the sorghum genotypes are going to vary. So for the methods, a lysometric-based experiment was set up in the greenhouse using 1 meter lysometric lysimeters, as shown here. The experimental design was a two-way factorial treatment structure running an RCBD. So two-way factorial involves, it means I used two factors. The first factor was the parental sorghum genotypes. There were 11 of them. These were genetically and then geographically diverse genotypes from different places of the world. And then the watering regime was at 2 levels. That is simply well-watered and then water deficit. So then I imposed water deficit stress for 15 days, starting at 35 days after emergence. Well-watered pots, I kept them at 100% filled capacity. And then the water deficit pots, I exposed them to 55 to 60% filled capacity. And then during the stress period, I used daily gravimetric pot weighing to impose the stress. And then to correct for evaporation, I had empty pots so that actual transpiration is obtained by deducting evaporation from evapotranspiration. So at the end of this stress period, the experiment was terminated, and agronomic traits collected included the tiller and leaf numbers, stem height and thickness, and shoot biomass. And then for the physiological traits, I measured the chlorophyll content using SPAD, gas exchange using Lycol, chlorophyll fluorescence using fluorometer, and then the leaf area using the leaf area meter. And then, as I noted at the beginning, working on the roots can sometimes be a bit difficult. So we took lots of strategies to get the roots out of the pot, and that involved one, rolling the pot on the ground, losing the the contents of the pot, and then secondly, we gently pour out the contents of the pot, and then carefully separate the roots from the growth media, which was surface. And then I lay it down, measure the length as an estimate of the rooting depth. After which then the roots were then washed, carefully washed. I store them in 20% alcohol. I take them out, cut them bit by bit, spread them in a film of water in a glass trays, make sure they don't clump together, and then I scan them to generate scan images. So on average, I was getting about 15 to 20 scan images per each plant or each root. And thereafter, the scan images were analyzed for various root morphological traits using the software WinRhizo. And then all data, both the shoot and the roots, were statistically tested using the GLM procedure in SAS. So some results here. For all the traits shown here, the shoot traits shown here, there was significant effect between the well-watered and then the water deficit treatment for almost all the traits shown here, regardless of genotypic differences. And similarly, there was genotypic differences for almost all the traits, regardless of the treatment effect. And then leaf number of tillers, photosynthetic assimilation, and stem height, there was genotype by treatment interactive effect. So based on that, I have some graphs to tell more of my story. On the y-axis above there is tiller number, below is leaf number. And then on the x-axis are the 11 parental sorghum genotypes that I used for my experiment. The blue bars represent the well-watered, and then the red bars are the water deficit. So this pattern is consistent for all the other graphs that I'll be showing in the experiment. So as Provara said, like if you're doing an experiment, try to relate it to the harvest index. So for instance, why tiller number? Because the number of tillers serves is indicative of the potential ability of the plant, because the tillers serve as a source of assimilate during the grain filling stage, because whatever is incorporated during the vegetative stage is able to be translocated for the grain filling stage. So the effect of the treatment for almost all genotypes resulted in less tiller number and the water deficit stress. And then if you look at the percentage difference between the well-watered and the water deficit treatment, we saw that genotype P898 had the least percentage difference, whilst genotype S1103 had the widest percentage difference, as can be seen here, where we saw the plants lacking tiller development whilst under stress. Similarly, we saw that genotype P898 had the least percentage difference for leaf number, and again, genotype S1103 had the widest percentage difference. And almost all genotypes had the least had less number under water deficit conditions. And this is basically because water deficit affects the initiation of new leaves and also adversely accelerate the senescence of old and mature leaves. Again, on the y-axis here, we have the photosynthetic assimilation, and we realize that the effect of the treatment resulted in less photosynthetic assimilation for almost all genotypes shown here. And this is due to the fact that water deficit stress induces the accumulation of abscisic acid, which results in stomata closure in an attempt to reduce water loss. But this, on the other hand, affects the conduction of carbon dioxide, which leads to less photosynthetic assimilation. Now, if we want to look at the percentage differences, we saw that genotype Marcia had the least percentage difference, whilst genotype SC2A3 had the widest percentage difference. And then transpiration efficiency here was obtained as a ratio of the shoot biomass to the cumulative water transpired, and realized that it was high for almost all genotype under water deficit stress. And most importantly, I just want to highlight on this genotype SC35, which had the highest transpiration efficiency under water deficit stress. And this is a noted stay green line, which has a post-flowering drought tolerance ability. And then for the shoot results, similarly for all the traits here, there were significant treatment effects. And then also for genotype, except that rooting depth was not significant. And then for the genotype by treatment interactive effects, that was total root length and then total root surface area. Like these were obtained from the scanned root images. So for the total root length, realize that genotype P898 again had, a significant higher total surface root length under water deficit stress, recording about -29%, and then genotype 1345 had the least percentage difference, although almost all the genotypes had a reduced in their total root length under water deficit stress. And then for the total root surface area, again, genotype P898 seems to do well, but not that significant. And so we saw that genotype S35 had the least percentage difference, and then genotype 1345 again had the widest percentage difference for total root surface area. So based on all the traits that we collected, some we could not represent here, the experiment was repeated and validated for 4 contrasting genotypes. 2 that did well, 1 intermediate, and then 1 that performed worse. And again, we saw similar results for almost significant treatment effect, genotypic effect, and also genotype by treatment effect for a few of the traits. And the same thing also for the roots, and again, we saw the rooting depth was not significant. So in conclusion, we say that yes, water deficit stress induced different water deficit tolerance traits, and sorghum genotypes varied in their responses to water deficit stress, which in a way confirms our hypothesis that I stated in the beginning. Sorghum parental genotypes such as SC35, Marcia, and then P898 can be considered as best performance under water deficit stress. And this research will provide pathways to map genomic regions for, which is responsible for increased resilience under water deficit stress. In that, we are collaborating with the Sorghum Genomics Lab in Kansas State so that they're able to focus on which population now to work on, because these are parents of sorghum mapping population. There are lots of them, and they can't decide to work on all the population, 250 of them. So at least this will help them to just focus on, let's say, SC35, and also the important traits also to focus on. And then quickly, something that I'm really excited working on, that is on the lipidomic side of things, because environmental stress such as water deficit stress affects the cell membrane. An important component of the cell membrane are the lipids. And so for my research, I took samples, leaf samples, I did some extractions to determine the effect of the stress on lipid composition. I analyzed them using mass spectrometry. And then a quick result here is a heat map, a snapshot of a heat map that I generated, which focuses on 2 important lipids, that is MGDG and DGDG. These are noted to dominate the chloroplast membrane, and as a result, they are very important for photosynthesis. So if we look up here, these are the 11 genotypes and the drought conditions, 11 genotypes and the well water conditions. And if we want to use the heat map color key, yellow means high, blue is low. So under water conditions, realize that these lipid contents are high, as we go to the deficit conditions, we realize that their contents are what? Reducing. And also some preliminary results from my calculations I'm doing, we are realizing that unsaturation levels is going down as water deficit stress is being imposed. So we are hoping that some results from this side will also help us to know what is going on at the cellular level, and then help us also to know which genes are regulating, and then we can identify and then help us to generate drought resilience crops. So on that note, I would like to say thank you to the crop physiology team in Kansas State, and a particular mention to the Kansas Lepidomics Research Center, and also for the student workers who have been helping me with my work. And that I will say thank you, and I appreciate any questions.
Any questions for Regina? Yes.
Okay, QT, that's what I look at. But especially when it gets to the genetic aspect, we collaborate with the sorghum genomics lab, and they are helping with that. But I currently have one lab mate who is working, has already started working on the populations using SC35, and he's trying to identify some QTS. So he's on that here. Okay, so the question is, I've measured so many traits, so which one do I think is good towards drought? Yes, that's an important question. I did some preliminary PCA analysis using the traits and then the genotypes, and yeah, for now I can't pinpoint, because we look at all the traits, because the fact that maybe one trait is doing well, maybe it's not doing well another, so we compare, check the percentage differences. But for me, one key trait I would say that at the vegetative stage, the shoot biomass is very important, because that's what translocates, and also transpiration efficiency. For me, these are the other two key things that I would say. But for now, like, it's difficult just to focus on one and say it's good. We look at all the others and check differences and see how it's doing well across all. I don't know if I've helped. The root what? Health. Okay. Okay. So. Okay. So the question is, from what she's read, she's expecting root surface, total surface area to increase under water deficit conditions, but then I had it be decreasing. And also, how did I measure, and did I even take into consideration the soil health? Okay. So when I started by saying I did not take into consideration the soil health, I think maybe I have, for instance, the soil microbiology lab. Maybe next time we can be an area to work, because I did not. So probably you are right, it could have an influence. And so how did I measure? As I said, I took them out of the pot, I washed, and then I scanned them. So then the software, WinRhizo, does the analysis and generates, tells me the total root length. But for the rooting depth, as you said, that one is just the length I measured, but just estimate the depth. But for total surface area, it's just by scanning and then analyzing using the software. And then on your third question, yes, I agree, because from some literature that I read, for instance, the rooting depth, it tells you that if plant is under water deficit, it's supposed to go deeper and then assess and like maybe put more energy and more resources into the roots here. But I think it wasn't exactly the case. But at least for one of my genotypes, as I indicated, P89, it realized was having higher total root surface area and then high total root length. So yes, it varies across genotype, and I was happy I got one of them, which can help for further research in my work.
Thank you so much, Regina.
Okay.